Hello my dear students, I am Parama Bhattacharya, Assistant Professor, Department of Food and Nutrition. Today I would like to discuss about blood and its composition. It is in the curriculum of your UG syllabus, mainly semester 1, and the paper is CC2T, that is human physiology. Okay, let's start. We discuss about our topic. Today's topic is blood and its composition. Okay, as we know that blood is one of the most important body fluid, and the color of that fluid is red, but actually it is not just like a fluid like other fluids in our body like tears, saliva, synovial fluids like that, but it is a very specialized living connective tissue. Okay, it is a very specialized living connective tissue, and initially I just summarize my content. Today's content is introduction, composition of blood and type of blood cells, function of blood, types of blood vessels and blood group. Now let us go. Okay. So, human blood is not just like a fluid, it is a very specialized living connective tissue, and it is also called as fluid of life actually, and it is consisting of different kinds of cells within the liquid matrix. Okay. And the total volume of blood in our body is approximately 5 liter, okay, approximately 5 liter, 4.8 liter to 5 liters, that is the 8% of our total body weight, 8% of our total body weight. Okay. Loss of 15 to 30% of blood in due to any kind of hemorrhage or like that, okay, pallor or weakness may precipitate, and if the percentage is more than 30%, loss of 30% blood may cause severe shock and death also. Okay. Blood is a fluid, obviously it is a fluid connective tissue, and that consists of mainly plasma, blood cells, different kind of blood cells and platelets, and it circulates throughout our body and deliver different important substances, okay, to various cells and tissues such as oxygen, different nutrients like glucose, plasma protein, cholesterol, different kinds of ions, iron, calcium, different enzymes, hormones, etc. Next, if we take the blood sample in a test tube with anticoagulant and centrifuge the sample, then we got 3 different parts of that sample. The topmost portion, that is the 55% of the blood, is mainly plasma, and the lower portion, that is 44%, is red blood cell, and in between the two layers there is a very thin layer, that is 1%, that is called buffy coat, right, buffy coat, that consists of mainly platelets and white blood corpuscles or WBC. Okay. So, the plasma, the percentage of plasma is 55% of total volume, and the formed element means platelets, white blood cells and RBC, that is the 1% and 44%. So, 45% of total volume is the formed element, okay, and the plasma is the liquid part of the blood, that is the clear straw colored fluid, right, that is the topmost layer of the test tube, and the plasma consists of liquid solvent, mostly water and solute, without the formed element. Okay, the solutes are without the formed element. Okay, 93% of water in the plasma and 7% is solute. Most solutes are protein like plasma protein, albumin, globulin. Okay, and the non-protein solutes are different salts, ions, gases, hormones, nutrients, waste materials of our body and enzymes, different types of enzymes, different kinds of enzymes. Okay. 8% of total blood is plasma protein, 8%, okay, and these are the albumin, globulin and fibrinogen. These are the three most important plasma proteins present in our blood. The most important is albumin. Albumin actually is responsible for the viscosity and osmotic pressure and blood volume. It is mainly responsible for the maintenance of viscosity and osmotic pressure of blood, and it helps in buffering. It helps in buffering and it also helps in transportation of many solutes, okay, by binding them like different kinds of drugs, pigments, fatty acid, bile salts, etc., etc. Okay, next one is the second most important protein is globulin. Globulins are some are antibodies. Okay, the antibodies are the part of our immune system, and some helps in transportation of some solutes, and some involved in clotting, in blood clotting mechanism. Okay, and the third most important protein is the fibrinogen, which is soluble precursor to fibrin, and it helps in the formation of mesh like structure or during the clotting mechanism. Actually the clot formation is fibrinogen helps in clot formation. Okay, and the next formed elements are the about 45% of whole blood, that is the erythrocyte or RBC, leukocyte and thrombocyte. Thrombocyte means platelets. All three formed elements are produced by stem cell. Okay, produced by stem cell. Now I would like to discuss the different formed elements of our blood. Blood consists of different type of cells which actually known as formed elements of blood. These cells have their own functions and roles to play in the body. Okay, the blood cells which circulates all around our body are as follows. Most important blood cell is red blood corpuscles or red blood cells, that is erythrocytes. Erythrocytes. Okay, erythrocytes are biconcave in shape. Okay, without nucleus in humans. It is very specialized cell and the structure is also very unique. The structure of RBC is very unique. Okay, and RBC contain the iron rich protein called hemoglobin, and the red color of RBC is due to the presence of hemoglobin in RBC, give the blood its red color. Okay, and RBCs are the most abundant blood cells produced in bone marrow. The main function is to transport oxygen from and to various tissues and organs of our body. Okay, the major function or the major job of RBC is to carry oxygen to cell to cell, and also deliver some carbon dioxide to lung. Okay. Next, erythrocytes or RBCs are biconcave disc, okay, with thin center and the thick edge. It is a biconcave shape. The center is thin and the periphery or the edges are thick. Okay, and the 7.5 μm is the diameter and 2.0 μm is the thickness. Okay, see the blackboard. I draw the structure of RBC like that. thickness is 2.0 μm, it is the thickness of the RBC, and the center is thin. Okay, the center position is thin rather than the edge of the RBC, the edge is thick. This is the beautiful structure of RBC, and this is the diameter, that is 7.5 μm. Okay, this is the structure of RBC, biconcave shape of RBC. Okay, it has the greater efficiency to carry gases, mainly oxygen and carbon dioxide. It has beautiful flexibility, and for this reason it can easily passes through very narrow capillaries of our body. It can easily passes through very narrow capillaries of our body. Okay, and RBCs are packed full of hemoglobin molecules. In each RBCs 200 to 300 million hemoglobin molecules are there, and each hemoglobin molecule can combine with 4 oxygen molecule, that is the oxyhemoglobin. Each hemoglobin molecule contain or can combine with 4 molecules of oxygen, that is the oxyhemoglobin. Okay. Therefore, each RBC can carry near about 1 billion of oxygen molecule. Each RBC, right? Each RBC can carry near about 1 billion of oxygen molecule. Next, okay, structure of RBCs, right? The plasma, the cytoplasm, the cytoplasm of RBC is filled with hemoglobin, okay, and which in turn contains acidophilia, causing erythrocytes to stain intense red color, okay, intense red color with eosine stain. When we stain the blood sample with eosine, hematoxin and eosine, hemoglobin, due to the presence of hemoglobin in the RBC, the color of RBC is intense red, okay. And the cell membrane, the membrane is lipid bilayer, okay, lipid bilayer containing 2 types of membrane protein. 2 important types of membrane proteins are there. One is peripheral and another is integral protein. Okay. The peripheral membrane proteins extend into cytoplasm and interact with an underlying cytoskeleton, which is mainly a meshwork of filamentous protein. The protein is spectrin. The spectrin is most important structural protein of RBC, and for the presence of this particular protein, due to the presence of this particular protein, the RBC is looks like biconcave shape. Okay, it is a very specialized protein. This interaction actually results in the characteristic biconcave shape of RBC and responsible for imparting strength and elasticity, of RBCs, enabling its even passage into the thinnest and smallest capillaries without any breakage. Okay, without any breakage, RBCs can easily passes through the very minute, thinnest and smallest capillary. Okay, and integral membrane proteins are innumerable, stretching throughout the thickness of the cell membrane, and it binds hemoglobin, serving as anchor protein. Actually, it acts like an anchor protein for the cytoskeletal network of RBC. Additionally, RBC express antigens, okay, on the surface of the membrane, cell membrane. They express antigens of ABO system, ABO blood group system, and erythrocyte surface antigens are necessary for blood transfusions. Okay. Next, most important point is erythropoiesis. Okay, erythropoiesis. What is erythropoiesis? Erythropoiesis is the process of origin, development and maturation. Okay, origin, development and maturation of erythrocytes. Erythrocyte means RBC. The RBCs are formed actually in our bone marrow from the cell that is called pluripotent hemopoietic stem cell, pluripotent hemopoietic stem cells, okay. And the stem cells are actually capable of self renewal process. They can have the self renewal process, okay, the stem cells. From pluripotent hemopoietic stem cells, colony forming blastocytes are formed, colony forming blastocytes are formed, and lymphoid stem cells are formed. From lymphoid stem cells is converted into lymphocytes and natural killer cells. Mainly the lymphoid stem cells are converted into lymphocytes and natural killer cells, and the colony forming blastocytes are converted into colony forming E cell, that is the colony forming erythroid, colony forming unit E, CFUE. Next, colony forming unit M, that is colony forming unit megakaryocytes, okay, colony forming unit megakaryocytes, and colony forming unit granulocyte and monocyte, okay. From colony forming unit M, that is colony forming unit megakaryocytes, platelets are formed. Colony forming unit megakaryocytes is converted into platelets, and colony forming unit granulocytes and monocytes are converted into white blood cells. But the colony forming unit E, that is the erythrocyte, colony forming unit E, that is CFUE, then again converted into erythrocytes, In the initial stage, the CFUE is divided into proerythroblast. Blast means rapidly proliferating cells, okay, proerythroblast. And these kind of cells have nucleus and immature nuclear chromatin, okay, 1, 2, 3 nucleoli is also present, and it is basophilic in nature. The cytoplasm is basophilic, okay. This is the proerythroblast, very rapidly proliferating cells, proerythroblast. Then the proerythroblast is converted into basophilic erythroblast. This kind of cell also contain nucleus, okay, and gradually the chromatin of the nucleus, the chromatins are gradually condensed, and nucleoli are absent, okay, and this is also basophilic. The cytoplasm is also basophilic, okay. After basophilic erythroblast, it is converted into polychromatophilic erythroblast. The chromatin in the nucleus are gradually condensed. There are 3 kind of changes in this process. One is cytoplasmic changes. Just see, gradually the color of cytoplasm is gradually changed from blue to violet to red color, okay. And cytoplasmic changes. The second one is nuclear changes, and the third change is structure. The structure is gradually reduces. The structure of the cell is gradually reduces, okay. So the first one is proerythroblast, the second one is basophilic erythroblast, third is polychromatophilic erythroblast, then orthochromic erythroblast, that is the much more dense nucleus, okay, with well hemoglobinized cytoplasm. Gradually hemoglobin is synthesized in the cytoplasm, and because of the generation of hemoglobin or the formation of hemoglobin in the cell, the color of the cytoplasm is gradually changes from violet to reddish, okay. Next one is reticulocyte, and after that the nucleus is ejected from the cell and reticulocyte is converted into mature RBC that is the red color mature RBC, and the nucleus is absent, and not only nucleus, there are other organelles like mitochondria, Golgi body, etc. are also absent in mature erythrocyte. Okay. Now, the next slide is red cell indices. Okay. Hemoglobin, PCV, that is hematocrit, MCV, MCH. Okay, these are the most important measurements of blood to diagnose any kind of diseases like anemia. Okay, hemoglobin is one of the most important parameter. By assessing the amount of hemoglobin in the blood, we can easily diagnose that whether the subject is suffering from, whether the patient is suffering from anemia or not. Okay. So, let us discuss. Blood is a mixture of plasma and cell, as we discussed in previous slide, and the packed cell volume, that is the PCV. PCV means or hematocrit value, যেটাই আমরা বলবো। PCV or hematocrit means the PCV or hematocrit test measures how much of blood consists of cells, how much of blood consists of cell. A PCV of 50%, PCV of 50% means 50 ml of cells are present in 100 ml of blood. Okay. If we take 100 ml of blood in a test tube and with anticoagulant, presence of anticoagulant, and we centrifuge the sample. After centrifugation, this portion is plasma and this portion is mainly the cell. Mainly RBCs are there. So, PCV, that is the packed cell volume, means the amount of cells in a packed form, okay, in a unit sample of blood, in a unit sample, that is the 1 ml or 100 ml or 10 ml, in a unit sample of blood, how much cells are present in a packed form, that is the PCV or packed cell volume. The percentage is 45%. In 100 ml of blood, if it is 100 ml, then the PCV is 45 ml. This is the PCV, packed cell volume. The amount of cell in a packed form in a given unit amount of blood sample. Okay. This is known as PCV. So, a PCV of 50% means 50 ml of cells are present in 100 ml of blood. If the number of red blood cells increases, if the number of formed element or red blood cells mainly increases, then what happens? The PCV also rises, it is obvious. It also increases as a result of dehydration. Okay. And the hemoglobin is the protein contained in the red blood cells that is responsible for delivery of oxygen to the tissues, and the amount of hemoglobin in whole blood is expressed in gram per deciliter, that is gram per dl, and the normal hemoglobin level for males is 14 to 18 gram per dl and for females is 12 to 16 gram per dl. Okay. And when the hemoglobin level is low than the normal value, then the condition is known as anemia. Okay. So, in 14 to 18 gram per dl means per 100 ml. Okay. In each RBC, 1/3 of RBC is occupied by, 1/3 of RBC is occupied by hemoglobin, right. So, 1/3 in each RBC, 1/3 of each RBC, 1/3 of each RBC is occupied by hemoglobin. So, if it is 45, if the PCV or the packed cell volume is 45%, then the 1/3 of 45%, right, the 1/3 of 45% in 100 ml of blood, 1/3 of 45% means it is 15, that is a 14 to 18, actually this is a 15 gram per DL, or 15 gram of hemoglobin in 100 ml of blood, because the each RBC actually contain 1/3 of hemoglobin. Okay. Next, wait. Okay. The next most important indices of red blood cell is mean corpuscular volume or MCV, mean corpuscular volume or MCV. MCV means PCV divided by number of red blood cells. PCV means packed cell volume or hematocrit value, that is the HCT or PCV, anyone. PCV means packed cell volume divided by number of RBC. Okay. If we know that 100 ml of blood contain how much amount of RBC, then we can easily calculate the MCV, that is the mean corpuscular volume. It is the measurement of RBC size. Okay. Next, the next most important indices is MCH, that is the mean corpuscular hemoglobin. Mean corpuscular hemoglobin, right, means the quantity of hemoglobin present in one RBC. The quantity of hemoglobin present in one RBC is known as mean corpuscular hemoglobin, and the next important indices is mean corpuscular hemoglobin concentration, that is MCHC, the concentration of hemoglobin in one RBC. The next one is the quantity of hemoglobin present in one RBC, and the next indices is the concentration of hemoglobin in one RBC, right. Okay. So, MCV means hematocrit by RBCs multiplied by 10, right. Then HCT means, or hematocrit means MCV multiplied by RBC by 10, MCV multiplied by RBC by 10, right. Then in MCHC, the formula of MCHC is, MCHC means concentration of hemoglobin in one RBC. Okay. Mean corpuscular hemoglobin concentration is hemoglobin divided by HCT, that is hematocrit, multiplied by 100, right. Then we can easily put the value of HCT. Therefore, hemoglobin divided by HCT means MCV multiplied by RBCs by 10, right. So, MCHC is equal to hemoglobin divided by MCV multiplied by RBCs and 110, that is the 1000. Okay. Then the formula of MCHC is hemoglobin divided by MCV multiplied by RBCs and 1000, multiplication of 1000. Okay. This is the formula of MCHC, that is the concentration of hemoglobin in one RBC. Okay, in one RBC. These are the most important indices of red blood cells. The next most important formed element or cell in blood is white blood cells or leukocytes. Leukocytes, okay. Leukocytes are colorless because it is devoid of hemoglobin, okay, and it is further classified into two broad categories, that is the granulocytes and agranulocytes, depending on the presence of granules in the cytoplasm, cell cytoplasm. And WBCs mainly contribute to immunity and defense mechanism. They actually the part of our immune system, and the size of WBCs are slightly larger than RBCs, and they are the only true cells which contain nucleus and organelles. WBCs are the only true cells of the blood that actually contain the nucleus and organelles. Okay. The main function is protection of our body because it is the part of our immune system. It can destroy the bacteria and pathogen and remove the dead cells and tissues of our body. And next, the different types of WBCs are, there are mainly 5 different types of WBCs and are classified mainly based on the presence and absence of granules. The first one is granulocytes. There are 3 types of cells, neutrophil, eosinophil and basophil in granulocyte. And in a granulocyte, there are 2 types of cells, that is the lymphocytes and monocytes, Okay. So, granulocytes are the leukocytes with the presence of granules in their cytoplasm, obviously, because it is the name is granulocyte. So, it is obvious that granules are present in the cytoplasm, and the granulated cells include eosinophil, basophil and neutrophil. Okay. Eosinophil are the cells of leukocytes which are present in the immune system, mainly immune system, and these kind of cells are responsible for combating infections, okay, and controlling the mechanisms associated with allergy and asthma, right, allergy and asthma. Eosinophil cells are small granulocytes which are produced in the bone marrow and makes 2 to 3% of whole WBCs. Okay, 2 to 3% of whole WBCs are made up of eosinophil, and these cells are present in high concentrations in the digestive tract of our body. The next important granulocyte is basophil. The percentage is very less, okay. They are the least common granulocytes, ranging from 0.5 to 1% of WBCs actually, and they contain large cytoplasmic granules, okay. And these white blood cells have the ability to be stained when exposed to basic dye, right, and hence referred to as basophilic, okay, because of it can stained with basic dye. And these cells are best known for their role in asthma and their result in inflammation, okay, and bronchoconstriction in the airways. And this kind of cells actually secretes serotonin, histamine and heparin, serotonin, histamine and heparin. The next most important and abundant WBC, that is the granulocyte, is neutrophil. They are normally found in the bloodstream and they are predominant cell which are present in pus also. Around 60 to 65% of WBCs are neutrophil, okay, with the diameter of 10 to 12 micrometers, and the nucleus is 2 to 5 lobed, multi-lobed nucleus, and cytoplasm has very fine granules are present in the cytoplasm of neutrophil, and neutrophil helps in the destruction of bacteria with lysosomes. And neutrophils are stained only using neutral dye, hence they are called so the name is neutrophil, okay. And neutrophils are the first cells of the immune system to respond to an invader such as bacteria or virus, okay. And next is the granulocytes. They are also leukocytes and with the absence of granule obviously in their cytoplasm, and agranulocytes are further classified into monocytes and lymphocytes, okay. Monocytes are usually they have large bilobed nucleus with a diameter of 12 to 20 micrometer, the size is larger, okay, and the nucleus is generally half moon shaped. The nucleus is half moon shaped or kidney shaped, and it occupies 6 to 8% of WBCs, okay, 6 to 8% of WBCs, and they are the garbage trucks of immune system, okay. They are called garbage truck, and the most important function of monocyte are to migrate into tissues and clean up the dead cell. That is why the name is garbage truck, okay. It can helps to clean up the dead cells and protect against blood-borne pathogens, okay, and move very quickly to the site of infections in the tissues, okay. And these white blood cells have a single bean shaped nucleus, okay, and hence referred to as monocyte. The next one is lymphocytes. They actually play a vital role in producing antibodies. Antibodies are very important component of our blood, and their size ranges from 8 to 10 micrometers. They are commonly known as natural killer cells, and they play an important role in the body defense mechanism, okay. And these white blood cells are colorless cells formed in lymphoid tissue and hence referred to as lymphocyte, okay, formed in lymphoid tissue and hence referred to as lymphocytes, and they are mainly of two types. One is B lymphocyte and the next one is T lymphocyte, and these cells are very important. B lymphocyte and T lymphocytes are very important in immune system and are responsible for humoral and cellular immunity. B lymphocyte is responsible for humoral immunity of our body, and T lymphocytes is responsible for cell mediated immunity in our body. Next most important component of blood is platelets or thrombocytes. Platelets or thrombocytes. Thrombocytes are specialized blood cell produced from bone marrow also. It is also produced from bone marrow, but it is specialized cell, but it is not whole cells or complete cells, okay. Rather it is a small irregular shape cell fragments of 2 to 4 μm diameters, right, and it is the second most abundant formed element in blood, and the range is mainly 150000, that is 1,50,000 to 3,50,000 per μl per microliter, okay. This is the range, and average range is 2,50,000 per microliter, right. Next, and the number of platelets in the blood, okay, varies depending on the site of collection, and no gender differences, right. And platelets come into play when there is bleeding or hemorrhage. The main role or main function of platelet is in the coagulation process, okay, and they help in clotting and coagulation of blood, and platelets help in coagulation during a cut or wound, right. The diverse functions of platelets are, it secretes vasoconstrictors, okay, and promote hemostasis and stimulate formation of clot dissolving enzyme, okay, clot dissolving enzyme to remove clots no longer needed, right. After that after the completion of clotting mechanism, the clot must be dissolved and removed, okay. Clots are not permanent. After repairing, the clot is removed by chemicals activated by the platelets, okay. Next, hemostasis, okay. Hemostasis, what is hemostasis? Hemostasis is the stoppage of blood flow, right, and there are three main mechanisms: vascular spasm, the first one, the second one is platelet plug formation, and the third one is clotting cascade. Okay. So, the first is the vascular spasm, right. When the arteries or the arterioles are damaged, right, the circulatory arranged smooth muscles in the wall contracts immediately. Okay, the reaction called the vasospasm initially contract. Okay, the smooth muscle in the wall contract immediately, and this is known as the vascular spasm, right. This reduces the blood loss, okay, and platelets release serotonin and thromboxane, and vasoconstriction occurs at the site of the injury and reduces the blood loss, right. This is the first step of hemostasis. Next, the second step is, the second mechanism is platelet plug formation, right. After 1 to 5 seconds, platelet plug formation starts after 1 to 5 seconds, okay, and adhere to exposed collagen fibers in damaged area, and to each other processes may stop if leak is sealed. Okay, the leak or the damage is sealed by the formation of platelet plaque. Okay, and the third point is blood clotting mechanism. Okay, if injury is extensive, clotting cascade is initiated. Okay, if the injury is extensive, then the clotting cascade is initiated, and it trigger the clump formation or the formation of clot at the site of injury, okay, and the fibrinogen is converted into fibrin, okay, and fibrin monomer is formed and stop the bleeding of the injured area, right. After the 3 stages, that is the vascular spasm, platelet plaque formation and the clotting cascade, what happens? Clot retraction, okay, clot retraction. After about 30 to 60 minutes, right, blood clot starts contracting, right, and when the tissue repair is completed and the clot is no longer needed, okay, then the platelets secrete factor 12, okay, activated factor 12, which stimulates the formation of enzymes and that dissolve the old clot. Okay, after the completion of whole process, the clot is dissolved, okay, and next is fibrinolysis. What is fibrinolysis? Lysis of blood clot inside the blood vessel is called fibrinolysis. The lysis of blood clot inside the blood vessel is called fibrinolysis. It helps to remove the clot from lumen of the blood vessel, right, and clot dissolution occurs continuously. Okay. Next, the differentiation between the plasma and serum. It's a very important area. What are the differences between plasma and serum? Plasma is transparent straw colored liquid portion of the blood which contain clotting factor, right, which contain clotting factor. But in case of serum, it is the extracellular portion of blood and which lacks clotting factor, right. This is the major differentiation, that is the plasma contain clotting factor but serum does not contain clotting factor. Okay. The next is, it is acquired after centrifuging blood with the anticoagulant. but it is acquired after centrifuging of coagulated blood. Okay, no anticoagulant is required. And plasma consists of 55% of total volume of blood, and serum less volume in comparison to plasma. Comparatively easier and less time is required to separate the plasma from the blood sample, but in case of serum it is difficult to separate serum from the blood sample. Okay, it is a time consuming process, and again plasma contains fibrinogen, is a very vital point, and the serum lacks fibrinogen. Plasma consists of 92% of water with protein, salt, lipids and glucose, and serum consists of 90% of water with dissolved hormones, proteins, minerals and carbon dioxide. Okay. And it has the density, that is in case of plasma 1.025 gram per ml the density, and in case of serum 1.024 gram per ml, the density is 1.024 gram per ml, and plasma has a long shelf life, okay, and it can preserved up to 10 years, okay, and in case of serum it has short lifespan but short shelf life, it can be preserved only for a few months. And plasma is the main medium for excretory products transportation. Plasma is the main medium, but in case of serum an important source of electrolytes only, it is an important source of electrolytes, okay. And in plasma cells are freely suspended in plasma, okay, but in case of serum cells are attached together by clot formation, okay. In case of serum the cells are attached together by clot formation, okay. Next is functions of blood. Blood is responsible for the following body functions. First is fluid connective tissue, okay. Blood is a fluid connective tissue composed of 55% of plasma and 45% of formed elements including WBC, RBC and platelets. And since these living cells are suspended in plasma, okay, and blood is known as the fluid connective tissue and not just fluid, okay, not just fluid. Blood provides oxygen to the cells, okay. Blood absorbs oxygen from the lung and transports it to different cells, different tissues, different organs of our body, and the waste carbon dioxide moves from the blood to the lungs and is exhaled. Transport hormones and nutrients. The digested nutrients such as glucose, vitamins, minerals and proteins are absorbed into the blood through the capillary in the villi lining, the small intestine, and the hormones secreted by the endocrine glands are also transported by the blood to different organs and tissues. Next, hemostasis. Blood helps to maintain the internal body temperature by absorbing or releasing heat, body temperature regulation, the another most important mechanism, homeostasis. It can regulate our body temperature. Blood clotting at site of injury. Next important role of blood, the platelets of the blood helps in the clotting of blood at the site of injury. Next, transport of waste to kidney and the liver. Blood enters the kidney where it is filtered to remove the nitrogenous waste materials, okay, out of the plasma. And next, protection of the body against pathogen, mainly the WBC or white blood corpuscles fight against infections. Next, the next topic is type of blood vessels, okay. Type of blood vessels. There are three types of blood vessels. These are arteries, veins and capillaries. Mainly there are three types of blood vessels in our body. Arteries are strong tubes and muscular in nature, and these blood vessels carry oxygen rich blood. Arteries carry oxygen rich blood from the heart to the cells of the body. Arteries carry oxygen rich blood from the heart to the different tissues, different organs of our body. Okay, and aorta is one of the main arteries that rises from the heart and branches further. Okay. Veins are elastic blood vessels which carry deoxygenated blood. Okay. Arteries carry oxygenated blood, but veins carry deoxygenated blood from all parts of our body to the heart, just reverse. Okay. Veins carry deoxygenated blood from the different part of our body to the heart, right. But an exception is the umbilical and the pulmonary veins are the exception. Okay. Next, the capillaries. On reaching tissues, the arteries are further divided into minute vessels or tubes called capillaries, right. Capillaries bring about the exchange of substances between the blood and the tissue. The main function of capillaries is to exchange the nutrients between the blood and the tissue. The next most important is sinusoids. They are the special type of wider capillaries present in the bone marrow, liver, lymph nodes, spleens, and some endocrine gland also. Okay. Next, different layers of blood vessels, right. Both arteries and veins mainly consists of 3 layers, right. The innermost layer is tunica intima. After tunica intima, tunica media, and the outermost layer is tunica externa. The tunica intima is the innermost layer. It is one of the innermost and thinnest layers of the arteries and veins. It comprises endothelial cells, and they are in the direct contact with the flow of blood, as it is the innermost layer of the arteries and veins. Tunica media is the middle layer of an artery or vein, and tunica media is made up of mainly smooth muscle cells. And tunica externa it surrounds tunica media, and it is made up of collagen mainly. It is made up of collagen, and it also supported by the elastic lamina in arteries. Okay. Now, see the differences between the arteries and vein, the structural difference in arteries and vein. Arteries carry mainly blood away from the heart, but veins carry blood towards the heart. Okay. And arteries are the wall of the arteries are much more thicker rather than the veins. Okay. The wall of the veins are thinner, but the lumen of the arteries are smaller, but the lumen of the veins are larger than the arteries, and the blood pressure of arteries are higher than the vein, and arteries have no valves, but veins have valves in their structure. Okay. Next another important area, that is blood group. Blood type or blood group refers to the kinds of antigen found on the surface of the blood cells. Okay. There are different kind of antigens are found on the surface of the RBC, and depending on the presence of antigen on the surface of the RBC, the groups or the types are actually determined. Okay. There are some protein in our body, that is self. Okay. All proteins and chemicals that are the part of our body, okay, are known as self, and non-self proteins are any proteins or chemicals that do not belong, okay, and antigens are the any foreign substance that enters in our body, and antibodies are some special some very special proteins made up of our immune system to remove some foreign substances. Okay. These are the basic concept, okay, self, non-self and antigen and antibody. Okay. Let us start. Actually, many antigens are present on the surface of the blood cells, creating many different blood groups. Only a few are important in blood transfusion. The most important system is ABO system and the RH system or research system. If during transfusion, okay, but before transfusion, if the blood group is not determined previously and the outcome or the result is agglutination, if the transfusion is mismatched, then the result is agglutination or clumping of the cell. It is very dangerous and it leads to heart attack, stroke, kidney failure, etc. Okay. So, before blood transfusion, determination of blood group is very important, most important thing before transfusion of blood. The ABO blood group system consists of 4 types of blood group: A, B, AB and O, and it is mainly based on the antigens and antibody. Antigens on red blood cells and antibodies in the plasma. Both antigens and antibodies are protein molecules in which antigens are present on the surface of the red blood cells and antibodies are present in the plasma, okay, which is involved in defending mechanism of our body. ABO blood group system, the basis of ABO blood grouping is of 2 antigen, mainly antigen A and antigen B, and the ABO grouping system is classified into 4 type based on the presence or absence of antigen on the red blood cell surfaces and the plasma in the and the antibodies in the plasma actually. In case of group A, contain antigen A on the surface of the RBC and antibody B in the plasma. Okay. In case of blood group A, antigen A is present on the surface of the RBC, but the antibody B is present in the plasma, mind it. And in case of blood group B, antigen B is present on the surface of the cell of the RBC, see the picture, okay, and the antibody A is present in the plasma, right. In case of group B, antigen B is present on the surface, cell surface, and antibody A is present in the plasma. And in case of group AB, contains both A and B antigen on the surface, on the RBC surface of the RBC, and no antibodies, neither A nor B, no antibodies are present in the plasma, that is neither A nor B, right. And in case of group O contains neither A nor B antigen on the surface of the RBC, but both the antibodies, that is A and B, are present in the plasma. Clear? So, in case of blood group A, antigen A is present on the RBC surface, but antibody B is present in plasma. In case of B, antigen B is present on the surface of the RBC and plasma contain antibody A. In case of group AB contains both A and B antigen on the cell surface, but no antibodies are present in the plasma. In case of O, neither A nor B antigen present on the cell surface, but both the antibodies, that is the A and B, are present in the plasma. Okay. This is the basis of the ABO group system. Next one is Rh factor or rhesus blood group. The Rh blood group is so named, Rh blood group is so named because the antigen was discovered in the blood of the rhesus monkey. Okay. And people whose RBCs have Rh antigens are designated as Rh positive. It is clear. The people whose RBCs have Rh antigen, whose RBCs have Rh antigens are designated Rh positive. Those who lack Rh antigens are designated as Rh negative. Okay? Clear? So, in addition to ABO system or ABO grouping, there is another system, that is the Rh blood group system, okay, which determine the negative or positive, that is A positive or A negative or B positive or B negative or AB positive blood group or AB negative blood group or O positive blood group or O negative blood group, right. If the rhesus factor is present, that is the rhesus Rh antigen is present on the RBC of the cell, then the individual is rhesus positive, means Rh positive, right. If the blood group of that person is A and also rhesus factor is present, then the blood group is A positive, okay. And if an rhesus factor is absent, then the individual is rhesus negative. If the blood group is A but rhesus factor is absent, then the blood group is A negative, right. And the compatibility of blood group during transfusion is very essential to avoid the agglutination, right. Okay. Thank you for listening.